All right, well then let's keep on going here to finish chapter 5. I'm going to skip ahead here. I was not prepared. We've already covered Sherrington's research on the nature of excitatory and inhibitory synapses, and we were leaving off right here to talk about exactly some more detail about how the synapse works at kind of a micro level here. We've already learned that during a nerve impulse and an action potential is generated along an axon, that this is an electrical signal. And that was discovered first, but after that, and after Sherrington had discovered sort of the characteristics of synaptic transmission, no one really knew exactly how the synapse worked, and especially the idea that there is a chemical secreted by the axon that travels across the synapse and stimulates the dendrite of the postsynaptic cell. This was not yet really understood, and we see here the discovery by Otto Loewy as late as 1921. That was 100 years ago, right? But a lot of this research on action potentials and axons began in the 1860s, so it took a few decades before neurotransmitters were first discovered, and this is the work of Otto Loewy here. So here I have a diagram that gives us a little hint of what Loewy had done. So what Loewy was doing here is he had a frog heart that was still attached to the vagus nerve. The vagus nerve regulates heart rate, and one of the neurotransmitters, which of course he did not know about it all at the time, was acetylcholine. And so what we now know is that the vagus nerve uses the neurotransmitter acetylcholine to slow down heart rate. And so let's look at the diagram here on the side. In one preparation that he had set up here, the frog heart is beating, and it stimulates using an electrode the vagus nerve, which will slow down the heart rate of this particular beating heart, right? And he collects the secretions from this heart, and he transfers those secretions to a second heart and notices that the second heart slows down as well, right? So curiously enough here, the idea is that the electrical stimulation creates the effect in this first heart, but in the second heart, it is not an electrical signal whatsoever. It is merely some chemical that has been secreted as a result of the electrical stimulation that creates the exact same effect, right? So he uses the electrical stimulation here as a way to get the first heart to produce a secretion, and then he demonstrates that it is this chemical secretion that has the real effect, right? He also later discovered then that epinephrine is another separate effect that speeds up the heart rate. And so he was able to transfer epinephrine chemically from one heart to another, causing the effect of speeding up the heart rate in that case. So whether we're using acetylcholine or whether we're using epinephrine, we get different effects, but they both show that the presence of this chemical has this direct effect on the beating heart. Now, of course, we have since learned about the role these neurotransmitters play in things like the parasympathetic and sympathetic nervous system to regulate cardiovascular output, but Loewy was the first to show that these chemicals, in fact, existed in the first place. And so now we start to understand that these chemicals, we're now going to call them neurotransmitters because they help transmit the signal between neurons, enable communication within the nervous system, both the central and the peripheral nervous system. We have also understood that these chemicals are essentially the same, whether we're talking about the nervous system or whether we're talking about hormones in the endocrine system. They are effectively the same kinds of chemical messengers. In a way, we can think of the brain as part of the endocrine system. It's a gland that produces hormones that can either work slowly to pass through the bloodstream and lymph to activate glands of the endocrine system, or they can work quickly within the nervous system, activating synaptic transmission that occurs very, very rapidly. A few facts here there are at least 50 different chemicals that have been identified as neurotransmitters. Some neurotransmitters tend to be excitatory. So as we talked about EPSPs and IPSPs, we know that some of the differences between those effects depend on the kind of neurotransmitter involved, right? So sometimes it's an excitatory neurotransmitter and sometimes it may be an inhibitory neurotransmitter. But to make things more complex, this is not an all or nothing sort of a thing. Some neurotransmitters can be either excitatory or inhibitory, depending on what part of the nervous system they are working in. So we have to also think about not just the nature of the neurotransmitter, but also the nature of the receptor site on the dendrite. And this is crucial because what we need to understand is that whether or not a given neurotransmitter has an effect on a postsynaptic cell depends on whether that postsynaptic cell has a receptor for it. And different receptors may do different things at the postsynaptic level. They may excite that neuron, they may inhibit that neuron, or they may do something else entirely. And so the nature of the postsynaptic response is something we're going to get into a little more detail as we go through these remaining slides. One little point here, again, at the very bottom here, is that this means that a dendrite on a given postsynaptic cell may have many different kinds of receptors that respond to many different neurotransmitters. So that means that at a given synapse, there may be many different neurotransmitters being released by the axon. This also occurs when there are many different axons all coming together. Remember the concept of spatial summation from Sherrington is that many different inputs, presynaptic cells, may be all converging their input to the same postsynaptic cell, and those different presynaptic cells may produce different neurotransmitters. So of course, the postsynaptic cell has to have different receptors for each one of those different transmitters. Here is a view of the synapse so that we can start to visualize what is going on with the neurotransmitter. Now we have here on the left this microscopic view of an axon terminal that is not too easy to really see there, but we see the what is labeled the axon presynaptic terminal. So presynaptic here is referring to the neuron that is sending the signal. That's the presynaptic neuron, and the one that is receiving the signal now is the postsynaptic neuron. And in that axon terminal, you see these little, if you can see my mouse, maybe this sometimes works in Zoom, sometimes doesn't. If you see my mouse pointer, all these little round, brownish round specks in the presynaptic terminal are called synaptic vesicles. And of course, we see it over in this drawing, which gives us perhaps a better view of what's happening there. These synaptic vesicles are labeled there as round granules. I like to think of them as bubbles, essentially, right? And they are bubbles that are full of molecules of neurotransmitters, right? And the can keep on going here. We see that there is a small gap between the presynaptic terminal and the dendritic spine that is referred to as the synaptic cleft, a very narrow space between the two. So the neurons here do not touch each other, right? There is a very narrow gap, that is the synaptic cleft. Now what happens, let's remember back to our action potential, right? The action potential is transmitted down the axon. And when that signal finally arrives at the axon terminal, this is going to trigger something else occurring, right? The axon action potential is going to trigger chemical changes and even electrical changes in the axon terminal that will cause these vesicles or bubbles of neurotransmitters to bind themselves to the tip, the surface of the presynaptic membrane. And we can see that down at the bottom here. This one particular vesicle has attached itself to the base of the synaptic terminal. And when it does so, it can now dump its contents into the synaptic cleft. And we see all those little red dots there spreading out into the synaptic cleft where they may come into contact with what are labeled here as the postsynaptic receptors, right? We can also see in this picture that there are not just little red dots, but there are also some yellow ones. So there may be some other neurotransmitters coming in from other neurons perhaps, or maybe that same presynaptic terminal is producing many different kinds of neurotransmitters. There's a few other things going on in here. We don't need to worry too much about the mitochondrion. That's the powerhouse of the cell, right? So, right. So there's just some metabolic things that may happen with the mitochondrion. But microtubules, right? So what are those? That's not really part of the action potential, but this is a transport mechanism that can transport certain chemicals from the cell body down to the to the terminal. And one of the important things we need to understand here is, where does the neurotransmitter come from in the first place? Well, sometimes neurotransmitter is manufactured right here in the presynaptic terminal, right? And that might be where some of these metabolic organs within the neuron are useful, is they can help manufacture the neurotransmitter and fill these vesicles full of neurotransmitter. But in other cases, neurotransmitter may be manufactured in the soma or cell body, and then it needs to be transported to the presynaptic terminal. And that's what the microtubules are doing, is they are transporting neurotransmitter from the cell body to the presynaptic terminal, where it can then be readied for use if an action potential happens to occur. Something else that we can also think about here is what happens after the neurotransmitter is released. Well, of course, I mentioned the postsynaptic receptors on the dendritic spine. So that is a signal that might activate the postsynaptic cell to fire an action potential. And then what happens is that we need to do something with this neurotransmitter. We don't want to leave it just sitting around in the synaptic cleft forever, because that might further cause additional stimulation of the postsynaptic neuron when that's not the intent. So we have to have various housekeeping operations that have to happen here to clean up the synaptic cleft to get rid of that neurotransmitter. And we will talk a little more about that the further we go into that. So here, in fact, is a sequence of events that will break this down. There's more than three. I have another slide coming up. First thing is that we have to make the neurotransmitters, synthesizing neurotransmitters. Most neurotransmitters are made out of amino acids, and we get amino acids in our diet. So oftentimes you hear about the so-called amine type neurotransmitters, right? And that is because they are made out of amino acids. We don't necessarily need to get into the chemistry of amino acids here, but that's just a fact that we'll need to know. As we are pointing out from that previous diagram, we're storing the neurotransmitters in the axon terminal, or we're going to transport them there, right? So they might be kept in the cell body and then transported to the axon terminal when we need them, or we store them in the terminal within the vesicles, right? The action potential, as it travels down the axon, will trigger the vesicles to bind themselves to the postsynaptic membrane. They are then dumped into the synaptic cleft. That process is called exocytosis, right? Exo meaning moving out, and cytosis referring to the cell. So moving out of the cell is exactly what this exocytosis means here. Neurotransmitters move out of the cell. Calcium also plays a role in this, by the way. So one of the other ions that we have been talking about, we've talked about sodium ions and potassium ions, chloride ions. I might have mentioned in the previous chapter's lecture that calcium plays a role as well, but I didn't say anything else about it. But here is the case here, is that there are channels, protein channels within the cell membrane near the axon terminal that allows calcium to enter into the neuron, into the presynaptic neuron, right? And the incoming calcium is what's going to trigger exocytosis of the neurotransmitter, right? Now, step 4. So the steps 4 through 7 here are going to finish us up. Technically, there's 8, depending on how you want to define step number 8. There's a picture coming up that has an 8 on it. I've simplified it to just 7 steps here. But step number 4 is that now that we have dumped the neurotransmitter into the synaptic cleft, it travels across the gap to activate the receptors on the postsynaptic neuron. And then that triggers a response in the postsynaptic neuron, and that response, of course, is going to be maybe an excitatory response. It fires an action potential. But meanwhile, like I said, we have to clean up the synapse. So what happens is that the neurotransmitters, after they attach themselves to the receptors on the dendrite, they then separate from those receptors, and now they're just floating there. So we have to clean it up, right? So there are different ways this can happen. Sometimes they are reabsorbed back into the presynaptic neuron. And this process is called reuptake, right? So we can have reuptake in which the presynaptic axon terminal sucks them back in and recycles them and prepares them to be reused. Sometimes they just diffuse away, right? There's just extracellular fluid and they just may float away into that extracellular fluid. Eventually, they might be cleaned up by astrocytes and then excreted into the veins where they are taken to the kidneys, and we will urinate them out, right? Sometimes there are other chemicals. So there are enzymes that break down neurotransmitter and break them down into other chemicals so that, again, they can be excreted. One example of this is called monoamine oxidase, right? MAO, right? And so MAO will break down the neurotransmitters into other chemicals that cannot have an effect on the postsynaptic cell. So they're no longer stimulating the postsynaptic cell, and then they can be cleaned away or sometimes recycled as well. You may have heard of certain medications that are called MAO inhibitors, right? So if MAO is something that helps clean up the synapse by eliminating neurotransmitters, an MAO inhibitor is something that prevents this from happening, which forces neurotransmitters to remain in the synapse a little bit longer. So if we wanted to do something medically through some medication to prevent the cleanup of the synapse, one way to do that is through an MAO inhibitor, which will then force the neurotransmitter to stay there. What would be the purpose of that? Well, maybe if we wanted to boost a particular neurotransmitter's action in the brain, one way to do that would be to allow it to just linger in the synapse a little bit longer. And by inhibiting MAO from breaking it down, that may allow that neurotransmitter signal to increase its strength a little bit there. Instead of having some of it broken down, you're just going to have a little more of that neurotransmitter in the synapse, activating more of those postsynaptic neurons. Now, one thing I also want to say about the receptors on the postsynaptic dendrite is that these things work like a lock and key system, right? So as I said before, that these different receptors can only be stimulated by specific neurotransmitters, and this has a lot to do with the molecular shape of the neurotransmitter. Different neurotransmitters are different molecules. Different molecules have different shapes to them, and only certain molecular shapes can unlock these receptors on the dendrites. And so when they're floating around, they encounter those receptor sites, and if they happen to have the correct molecular shape, they lock into that receptor, and then they unlock it, which opens up a door essentially into the postsynaptic cell, right? Now, if they don't fit, then they just kind of bounce off, right? They don't really have an effect there, right? So they have to have the right shape in the same way that a key has to have the right shape to unlock a door. Here, the neurotransmitter has to have the right shape to unlock these receptors. So these receptors really are doors. They are gateways. They are protein channels. And as we talked about in the previous chapter with action potentials, when we open up a channel, some kind of chemical, a positively charged ion is going to either go into the cell or out of the cell. And in particular, these are sodium channels, right? So these receptors, when they open up, they only let sodium through. And so we're going to let a little sodium into the cell, which is going to raise the internal voltage if this is an EPSP, right? And then what happens, of course, is that if it's a strong enough influx of sodium that raises the internal voltage of the neuron to its firing threshold of negative 50 millivolts, perhaps, then that's going to trigger the action potential. But of course, this is where those graded potentials come in. How many receptors are being stimulated? And do we have some temporal summation? Do we have any spatial summation? If we get a strong enough influx of sodium to raise that internal voltage, we're going to see that action potential. But again, of course, what we find is that some neurotransmitters are inhibitory. The receptors might actually be potassium channels, right? And if that opens up a potassium channel, that lets potassium enter the neuron, which actually, that lets potassium leave the neuron, right? And that's what's going to happen because of the concentration gradient. A little potassium might leak out, and that's actually going to further lower the membrane potential below its resting of negative 70, which would create an IPSP. Right? Now, eventually, because the postsynaptic cell has received some stimulation from this neurotransmitter, the postsynaptic cell also has this ability to communicate backwards to the presynaptic cell to say, okay, I received your signal, it's time for you to stop, right? And that's what's happening here is that a signal going from the postsynaptic cell back to the presynaptic cell is basically telling the presynaptic cell, signal received, now stop doing what you're doing. So it will stop, the presynaptic cell will actually stop releasing any more neurotransmitter, and instead we'll go back to the cleanup process to help eliminate any neurotransmitter that remains in the cleft. And then everything basically resets and goes back to ready to do it again if it needs to happen, right? So this picture shows all of those steps 1 through 7, plus an 8. That's, like I said, we can break out the final step into 2 parts, which is what the way it's labeled here. But let's go back to step number 1, right? We have to manufacture and synthesize the neurotransmitter. So we see that broken out as 1A and 1B, because in some cases it is synthesized in the cell body, which is what 1A is showing us, and 1B shows us the synthesis of other neurotransmitters in the axon terminal. In many cases, it has to do with the size of the molecules, right? So a small neurotransmitter means a small molecule of neurotransmitter, such as acetylcholine, right? Whereas other neurotransmitters might be manufactured in the cell body, and then transport occurs. So transport occurring in step 2 is through those microtubules. We're taking it all the way down the axon. That's not an action potential, right? This is just transport to the axon terminal. And again, we can store everything in these vesicles here. So now we have step 3. If an action potential occurs, we end up with exocytosis. So here we see at step 3, the vesicles bind themselves to the terminal button cell membrane. They then dump themselves into the synaptic cleft. And again, calcium plays a role in triggering this exocytosis. In step 4, we see those little red molecules of neurotransmitter binding to the receptor sites on the dendrite of the postsynaptic cell. In step 5, we see them separate and begin to float off to the side. Step 6, we're going to start cleaning them up. So we see in this case reuptake. We see some of these neurotransmitter molecules re-entering the axon there at step 6. Steps 7 and 8 show the feedback loop. So what happens here is that at 7, the postsynaptic cell releases what's called a retrograde transmitter. So this is also a chemical, a neurotransmitter in a way, but it's called retrograde because it's going backwards now. It's sending a signal from the postsynaptic cell back to the presynaptic cell to tell that presynaptic cell, stop doing it, right? Shut up now, right? Stop sending any more neurotransmitter. And that's what 8 is showing us here that I didn't list on the previous slide, is that 8 is the actual receptor on the presynaptic axon that receives the retrograde transmitter, right? So 7 is just the production of the retrograde transmitter, and 8 is receiving that signal that says, okay, yeah, I'll stop making any more neurotransmitter, or I'll stop releasing more neurotransmitter. What's also showing here, a bit of an unusual drawing, is this green thing wrapping around the synapse, which is part of an astrocyte, a glial cell, right? And so the glial cell can be where we get this monoamine oxidase, right? That produces this enzyme that cleans up the synapse and helps absorb extra used up neurotransmitter and other chemicals, so that we can get rid of all of that stuff. So the housekeeping job of the astrocyte, we see that here. So again, what are the kinds of synaptic responses, the postsynaptic responses? Well, as I have already said, in some cases, the receptors may be sodium channels, in which case that will allow positively charged sodium to enter into the neuron. That's going to raise the internal voltage, a graded potential, and that's depolarizing, right? We are getting closer to 0 with the internal voltage, right? And that would be an excitatory response, or EPSP, if it crosses the threshold, right? And then we will see an action potential. This is called a type 1 synapse. Type 2, may be obvious now to guess, is an inhibitory response. So these are the hyperpolarizing effects. So this is when those postsynaptic receptors might be potassium channels. So we open up a potassium channel that lets a little positively charged potassium exit the cell, which lowers the resting potential, or the membrane potential, below -70, making it harder to fire an action potential now. This is a type 2 or IPSP synapse. As it turns out, there's even a third kind of synapse. Besides the excitatory type 1 and inhibitory type 2, there are others that cause a different kind of chemical reaction that has other effects. What are the other effects? Well, let's move on a little bit here, because another way to break down these types of receptors and types of synapses is to take type 1 and type 2 and give them another broader name called ionotropic receptors. So what is an ionotropic receptor? Well, it is a receptor that does something with ions, right? So the sodium channels involved in the EPSPs, that involves, the sodium ions entering into the cell. The potassium channels are the ones that let potassium out of the cell to do the inhibitory type 2 effect, right? Glutamate, for example, and acetylcholine, that's the ACH abbreviation there, are typical examples of excitatory neurotransmitters that tend to activate type 1 synapses. GABA is a different neurotransmitter that has mostly an inhibitory effect in the brain, so it stimulates lots of type 2 synapses. But these are all ionotropic because they have an effect on ions and how ions influence this neuron, right? Either entering or leaving the cell. But then that third kind of receptor and synapse is called a metabotropic receptor, right? So what is a metabotropic response? That's one that changes the cell's metabolism, right? So that's what we have described here, changing the cell's metabolic activity. What happens is known as a second messenger signal. So a second messenger here is that when the metabotropic receptor is stimulated, this triggers a separate set of chemicals called second messengers, because they are now not part of the ion channel here, but they're internal messengers inside of a cell that trigger metabolic responses. And what are these metabolic responses? Well, these are changes in protein functions inside of the cell. And what they can do, for example, is they can cause that cell to maybe start making certain neurotransmitters, or increase how much of a neurotransmitter it makes, or even decrease how much of a neurotransmitter it makes. What it can also do is it can also trigger building new ionotropic receptors or ion channels, right? So that means maybe we want to build some more sodium channels. Maybe we want to build some more potassium channels, or maybe we want to eliminate some sodium channels, which can also happen, right? So in this case, the idea is that if we increase the number of sodium channels, that possibly will make, think about what happens here, right? If sodium coming into the neuron is something that works as a EPSP to stimulate the action potential, it would make sense that if we have more sodium channels on the dendritic spines, that this could actually cause that postsynaptic cell to be more sensitive, right? Because the more sodium channels we have, the more likely it is that these channels open up and let sodium in, and the more sodium that comes in, the more likely that we are going to have an action potential in the postsynaptic cell. That's possibly a metabolic process that could occur, is that if we wanted to increase a particular cell's sensitivity, we want it to build some new sodium channels. If we want to make it harder to stimulate this cell, we can maybe eliminate some sodium channels, or we might add some type 2 receptors, right? Things that effectively inhibit that neuron. So if this particular neuron happens to grow a bunch of type 2 receptors, then suddenly it's going to be easy to inhibit this neuron rather than easy to excite the neuron, right? And there might be various reasons why we want a particular neuron's sensitivity to excitation or inhibition to change over time. The ability of the brain to change over time is an important part of learning and storing new information in memory, is to change neural patterns over time. And the way this happens is through these metabolic processes. It's not just about activating synapses all the time, but sometimes it's about changing the strength of a synapse over time to either sensitize that synapse to make it easier to activate, or to desensitize the synapse to make it harder to activate. And it's these kinds of slower changes in synaptic function that are also a very important part of overall brain function and learning and memory and all this stuff. All right, so in this particular case, we don't need to linger on this picture very much. I threw some pictures in here just to help visualize some of the processes, but this captures what I said before about neurotransmitters working like a key in a lock, right? And so we see the molecules here, and we see a little bit of a magnified view of the keyhole there, this little kind of semicircular little concavity on the external surface of the receptor. And in the case of these red ones, those are neurotransmitters which fit into the binding site. They bind onto it, right, and unlock it. And the result of that, as I said before, is it literally just opens up a door, right? It's not just a receptor. It is a ion channel, right? And it is closed, except when the neurotransmitter binds to that receptor site, and then it opens up, and then we see some ions passing through into the neuron. In this case, this would likely be a sodium channel that results in an integrated EPSP. Let's have a look at some visualization that occurs here with some metabotropic receptors. Now, there's a little bit of detail here with some of these proteins that I had mentioned here that we don't necessarily need to worry about too much, like what these proteins are and where they come from and how they work, having alpha units and beta units and gamma units and stuff like this, or G proteins, I should say, but and how these subunits and messengers really work. But I just want to give you the idea here that here we see on the top left, again, the neurotransmitter comes in, it binds to the receptor, but instead of opening up a channel, what it does is it triggers the release of that G protein from the bottom or the inside of that receptor. And that G protein then travels around inside of the neuron where it might do some things, right? It can activate enzymes and it can do things that will then form new channels, or it might even open channels from the inside. That's another thing entirely that I didn't mention before, right? But sometimes these ion channels are opened from the outside by neurotransmitters, but sometimes they get opened from the inside through proteins that are activated through these metabotropic receptors. But more often, these metabotropic receptors are playing an important role in changing the metabolic function of the neuron to, like I said, synthesize more or less neurotransmitter, or to synthesize more ion channels and so forth. So these are growth processes, really. There's a good way to think about it. The other thing we want to talk about here to finish up this chapter is to think about the different kinds of neurotransmitter systems that exist within both the central and peripheral nervous system and throughout the endocrine system. So we have here, remember some of our breakdowns in the peripheral nervous system. We had the somatic nervous system, which involves sensory and motor nerves. Here we have what's referred to as the cholinergic system. This is the set of neurotransmitter processes involving the use of choline or acetylcholine as the primary nervous transmitter. So where do we find receptors for acetylcholine, we would then call them cholinergic neurons if they contain receptors for acetylcholine? Well, we find them in the muscles, also in the motor neurons that are used in the spine to stimulate the muscles, right? So acetylcholine is excreted from the axons of those motor neurons, and the direct application of acetylcholine to muscle fibers, the muscle fibers themselves actually have receptors in the same way that dendrites have receptors. And then once they are stimulated by acetylcholine, they will contract. So that's what we refer to here as the nicotinic ACH receptor, or the actual receptors on muscle fibers and sometimes other neurons that are directly stimulated by acetylcholine. Otto Lowy's research with the frog hearts, right? The heart is made out of muscle as well, right? It's not skeletal muscle, it's smooth muscle. But of course, acetylcholine will have an effect on that muscle as well. Now, in the autonomic nervous system, remember we is subdivided into the sympathetic nervous system, which is the fight or flight response, and the parasympathetic rest and digest response. So as we previously mentioned with Lowy's discoveries here, norepinephrine is the hormone / neurotransmitter that is excitatory on the heart, but also, curiously, inhibitory in the digestive system, in the gut. Acetylcholine is the opposite, right? It is now inhibitory on the heart and excitatory in the gut, right? So we just think about how that's going to work, is that the receptors on the heart are basically type 1 receptors that are norepinephrine or noradrenaline. So sometimes we call them noradrenergic receptors, but they are type 1 receptors on the heart for noradrenaline. In the gut, receptors for noradrenaline or norepinephrine are type 2 receptors, right? Inhibitory. We see the converse thing happening with acetylcholine. Now, why is this the case? Because, of course, what we said before about the fight or flight response is we want to increase energy right now in the moment. So what we want to do is we want to increase the cardiovascular output to get more nutrition and oxygen flowing to the muscles. So we want to increase heart rate, but we don't want digestion because digestion is really very slow as a way of getting energy, and that's not necessary during a fight or flight response. So we're going to inhibit the gut. But of course, when we're not in a fight or flight high stress situation, we're going to let the parasympathetic nervous system take over. So in order to rest and calm down, we use acetylcholine to slow down the heart rate and calm down the cardiovascular response, and meanwhile, re-stimulate digestion. Now let's move back into the central nervous system. So we cover the basics of the peripheral nervous system with those neurons. In the central nervous system, we again find a cholinergic system, right? So just like we see cholinergic neurons in the peripheral nervous system, we find them also in the brain. But here in the brain, these play an important role. They are excitatory for the most part in the brain. They play an important role in memory formation. So when it comes to encoding new memories, we need to invoke neural changes. So we see the cholinergic system is important for that. There are dopaminergic neurons. So this uses the neurotransmitter dopamine. We find a lot of the receptors for this in the limbic system. So remember, the limbic is the emotional part of the brain. The basal ganglia is part of the movement-related part of the brain, and also in the brainstem, down into the midbrain especially. And what we find is that dopamine plays an important role, for example, in the control of movement, which would make a lot of sense if the receptors are found in the basal ganglia and midbrain. We may have talked a little bit about Parkinson's disease when covering these parts of the brain before, and the substantia nigra that is found in the midbrain and with projections that also synapse up into the basal ganglia. That damage to that part of the brain is oftentimes implicated in the movement tremors that start to occur in Parkinson's disease, and also even in Tourette's as well, with the unwanted movement tics. As it turns out, dopamine is the primary receptor on these neurons. And so this is a dopaminergic pathway here. And a lot of the drugs that are used to treat Parkinson's disease involve trying to increase the dopamine signaling here. When those neurons are degrading, that means the signal is weaker, and there's less. And so if we increase dopamine, we might be able to magnify that response a little bit. We also see dopamine playing an important role in learning and reward, right? So this idea of sometimes we call the dopamine reward system is that anytime we achieve a goal and have some satisfactory outcome, this is associated with activation of dopaminergic neurons from the brainstem and midbrain and up through the frontal cortex, actually. We also have a noradrenergic system. What's the difference between noradrenaline and norepinephrine? Not much. Sometimes we just talk about them saying, use different words for whether we're talking about central or peripheral nervous system, for example. Where we find the noradrenergic system? Well, in the cortex and also in the limbic system. So it makes a little bit of sense. If this hormone is used as part of the fight or flight response to increase cardiovascular output and inhibit digestion, that hormone / neurotransmitter should also affect the emotional centers of the brain to maybe activate the amygdala to help let us know, oh, I should be afraid, I should be stressed, I should be feeling some emotion to go with this physiological response. But it also plays some role in learning as well. We're not going to get into all of the details of how learning changes neurons quite yet, right? But that's coming up. And then also the serotonergic receptors, right? So this is for the neurotransmitter serotonin. We find them in the brainstem and midbrain. The Rafe nuclei, which we haven't mentioned yet, are actually part of the reticular formation, which we previously saw is a network of nerve fibers that span from the brainstem up through the midbrain that are part of the arousal centers of the brain that help regulate our circadian rhythms of waking and sleeping. And so serotonin does in fact play an important role in our circadian rhythms. You might have heard of another neurotransmitter called melatonin, right? And melatonin is actually a derivative of serotonin. And so Melatonin helps regulate, serotonin is broken down into melatonin. That's part of the job of the pineal gland, and that helps regulate our sleeping and waking cycles. All right, that is everything for this chapter. So that's it.